Okay, we're recording, so thank you very much, William, for agreeing to give a Build a Cell seminar, and it's all yours.
Hello, everyone. I'm William Poole. I am a final year graduate student at Caltech, co-advised by Eric Winfrey and Richard Murray, and I've been involved in Build a Cell since the start. And today I'm going to be talking about a software project called Biocompiler. So let me share my screen. Great, that looks like it's there. So this talk is called Compiling Synthetic Biological Circuit Models with Biocompiler. And although I'm talking about synthetic biology models, this could also be used for systems biology and any type of biochemical model, or many types, not any type. And I want to just from the get-go say this is not all my work. There's been a team of us who've been doing this software, including Ayush Pandey, Andre Shore, Zoltan Tusa, and my PI Richard Murray, who has been very supportive. So what is Biocompiler? I think the best place to start is with the name. So first you pronounce it Biocompiler, not BioCRNpyler, although you could say that if you wanted to, I suppose. But let's sort of break this apart for a second. Bio is there because it's a biological or software for biologists. It is a library of biochemical components and mechanisms. And I'll explain what components and mechanisms are specifically, but I think you can imagine what this might be. CRN stands for chemical reaction network. And this is a mathematical modeling framework that I think many of you should hopefully find familiar, or maybe you're already experts in, but it's a very good way to represent what we know about biochemical models, things like gene regulation. Compiler is something from computer science. It's the idea of taking a simple specification to generate something more complicated. In the case of Biocompiler, we're using simple specifications to generate complex systems of equations that represent biochemical systems. And finally, the PY is for Python. Python is a programming language many of you have hopefully heard of, and we decided to make Biocompiler in Python because it makes it easy to plug this piece of software in with other tools and pipelines. So we're not hoping that Biocompiler solves all of your problems you ever have as a computational biologist or a experimental biologist, but rather that it is one tool you can easily use and easily connect it to other tools you also already use. So this is a modeling talk, and before going into details, it's good to give an idea of how I think about modeling in relation to biology and bioengineering. So the first way I think about modeling is from an engineer's standpoint, where I think of models as designs for biochemical systems. If I'm a synthetic biologist and I want to build a system, I might use a piece of software, maybe like Cello, to design some logic gates that have the properties I want. I might use software like Biocompiler or sub-SPML to produce a model of that system. I might use a simulator. BioScrape is the simulator that has from the Murray lab that is used for all the simulations you see in this talk. It's connected to Biocompiler, but is by no means the only simulator. There are many simulators out there. To simulate that system, maybe I do some numerical analysis to learn something about it, or maybe I do parameter inference to connect my model to data and understand, parameterize my rate constants based upon data I've collected. And that brings me back to the design. I might say, oh, my design system, when it's simulated and compared to data, doesn't seem to be working. Maybe you have to go add a new module to correct for some experimental behavior I didn't take into account. And you can repeat this process iteratively until you get what you want. You can also use models as a biologist to test hypotheses in silico before doing expensive and slow experiments. So when you think about sort of a modern biology lab, especially with omics data and automation, it's very easy to collect huge amounts of data, but it's also time-consuming and expensive. You can spend $10,000, $20,000 sequencing DNA and get a huge amount of data, but you want to do that carefully. And models help you do that. So given some data, you can try to build models to better understand that data. And in particular, models codify hypotheses. So by writing down a model, you're saying, I think this is how things work. This is my hypothesis, and Biocompiler lets you do that. Once you have a model, you want to connect it to the data. You want to ask, does our model explain my data? Does And parameter inference is a way of finding parameters for your model from your data. And I'm not going to go into it, but once you do that, it's pretty easy to ask, does my hypothesis explain my data? In other words, do they look similar quantitatively or qualitatively? And even, is the data informative enough for the model? So what I mean by that is, in many cases, you write down a very, very complex model, but you have one fluorescent protein, and your data is not informative enough to test your hypotheses. And so rather than going back and collecting a whole bunch more data, you can, which is slow and expensive, you can go back and, play around building some models and comparing them to your data to see what your data can actually teach you and what hypotheses fit your data. And that will help guide the next round of data you collect. And where does Biocompiler fit into this all? It is a tool for building and deploying models. So let's take a step back and start with, chemical reaction networks. This is the modeling framework that Biocompiler uses. And they're very common in synthetic and systems biology. And, you probably saw some of them in high school, even in your high school chemistry class. But we're going to take a more formal approach at first before giving some examples. So formally, a chemical reaction network is a set of species. Here I'm calling them X1 through Xn, and a set of reactions. Reactions take a multi-set of input species and at some rate or propensity produce output species. And there's a whole network of these reactions. So there's many reactions with many different rates and many sets of inputs and many sets of outputs. Now, the thing I really like about chemical reaction networks is they're both abstract and expressive. They can represent information in many different ways. So what I mean by that is that species and reactions don't really have to be things about atoms necessarily. They can be more abstract. For example, in high school chemistry, you might have seen a chemical reaction like this, the burning of hydrogen to produce water, where here my species are actually molecules with atoms enumerated inside of them that change the chemical bonds. In molecular biology, you might think of a chemical reaction like this, where a gene produces a gene plus a transcript. Now, this is not enumerating all of the carbon atoms or all the nucleotides even. This is a much more abstract view of what the process of transcription looks like. But in many cases, this is good enough to produce a model. And the math underlying this very simple gene goes to gene plus transcript is the same as the math you would use to model the burning of hydrogen. So that's very convenient. And you can get even more abstract and think about populations of things. Here I can imagine that I have a black and a white bunny, which come together to produce 5 gray bunnies. And this is also a chemical reaction network, or maybe in ecology, sometimes people call them Petri nets, but it's the same fundamental principle where I have things coming together to produce more of different or the same thing. Now, I'm not talking about individual atoms very much in this talk, and I'm not talking about bunnies or populations of organisms. Instead, I'm talking about molecular biology. And there's some motifs in molecular biology that we come across a lot. So for example, 2 molecules might bind together. And I wrote this as a reversible chemical reaction on the left. And on the right, I sort of have a little picture of what that might look like, where the half circles come together to form a full circle. And this is a motif that you'll see throughout the talk, that we often have things that bind together, and this is how I represent it. Catalysis is something else that's very important in biology. So I've written 2 different ways of representing catalysis. A plus C goes to B plus C, where C acts as a catalyst, or you can have A and C bind together and then produce I'm supposed to say C and B. I have a typo there, I'm sorry. You can also have compartments. So imagine I have, an external signal and an internal signal, and it diffuses across a compartment. Well, I can also call that a single step reaction, where the signal goes from A internal to A external. And finally, conformations. Many biomolecules have different conformations or states. So maybe you have an open state, and then a transition state, and then a closed state, and you can transition between them. All of these things are easily expressible as chemical reaction networks, and this makes the framework very powerful for codifying our knowledge and how, our internal picture of how biochemical systems function. So once you have a chemical reaction network, I've explained what they are, but I haven't really explained how we get numbers or simulations out of them. So I'm briefly going to go through that. There's two types of dynamics. The first is deterministic. And so in deterministic dynamics, I've written the same thing here where I have my species X and my reactions are written in sort of the vector form. So here I have the inputs is given as a vector, the number of inputs I of each species xi, and the outputs are given as a vector as well, and my propensity function. And I can write down this set of ordinary differential equations. The change in x with respect to time, it's a concentration right now, is given by the sum over all the reactions of the difference between the inputs and outputs times the propensity. So this is a fairly compact ordinary differential equation, but there might be many species and many reactions. So in fact, although I wrote this in a very compact way, typically this has many terms and is very highly dimensional. But let's give a very brief example. So here are three reactions. I have nothing goes to A and A goes to nothing, and I've written the rates above and below. And then I have a catalyst converts A into 2B. And I wrote the rate here. So I'm going to start. C is a catalyst. It never changes. Its inputs and outputs are always the same. So dc dt is 0. B is produced. There's only one reaction to produce it. So its rate of production is 2 times the amount that's 2 times B from over here times the rate, which is a function of A and C. And finally, A is involved in 3 reactions. It's produced from nothing at a rate 10, so that gives me a constant. It goes back to nothing at a rate of 2A, so minus 2A. And then it's also consumed in this second reaction at a rate of CA over 3. So that's negative. And I don't have to be able to solve this set of equations by hand. I can just plug it into a computer and integrate it to get trajectories. And you notice that C is constant at the bottom, B is produced, and A is degraded. Now, that's deterministic dynamics in a nutshell. I'm also going to briefly mention stochastic CRN dynamics. One thing I like about chemical reaction networks is that the same network can give rise to multiple mathematical interpretations. And in this talk, though, I'm not really going to talk about stochastics at all, but I'll note that everything I say about Biocompiler works both for deterministic and stochastic CRNs. And so in the stochastic world, instead of having a set of ordinary differential equations to describe your concentrations, you use something called the chemical master equation, which is also a set of differential equations that describes a Markov chain on the integer lattice. Now, that's a lot of math terminology, but I think you can break it down pretty simply. Instead of having concentrations of molecular species, we're going to talk about counts. So now instead of having 2 molar of A or 2 millimolar of A, I'm going to say I have 2 molecules of A, or I have 10 molecules of A. So I'm actually counting the number of molecules. And instead of having continuous changes of concentration where my concentration is changing, I'm actually going to change the probabilities of counts. So we can kind of see that in these pictures. The top picture, the dotted line is showing me the deterministic simulation. And then these solid lines are a whole bunch of stochastic trajectories, which are always integers. You'll see that you'll have a constant number, then it jumps to a different value. So that's called a Markov jump process. And that every trajectory is different, right? So I'm showing many different trajectories and they're all different. But on average, the trajectories of the stochastic simulation are the same as the trajectories of the deterministic simulation. So you'll see that this red line is right in the middle of the red ensemble of trajectories. Now, if I think of this as a distribution, so now here instead of saying, okay, here's what one simulation or one experiment might look like, I can say, what does the probability distribution, how does my probability change with time across this ensemble? And that gives rise to this sort of probability distribution. Here you see darker means when I always start at 1 in the simulation, and I get lighter means less probability, and then my sort of probability diffuses to some steady state. Or on the case of B, you'll see that we're always, on counts of 0, 2, 4, these even numbers because of our reaction. So we have a probability distribution, but it's over a different space. And this, I think, is just good to know that you can have both these interpretations of a CRN. Now, I've shown that CRNs can sort of model stuff, deterministic concentrations and stochastic counts of species. What can they not model? I think this is very important to know if CRNs are the right thing for your system. They can't model locations of molecules in space. So CRNs are fundamentally discrete. I can have a finite set of states or a finite set of locations, but if I have continuous motion, like diffusion in a box, I can't model that. Maybe I can model diffusion on either side of the box. I can be on the right side or the left side, but I can't be at any given point in the box because that's a continuous variable and CRNs are discrete. I also can't model detailed chemical structures. So if I'm interested in the detailed conformation, the continuous locations of all of the molecules in an alpha helix or another structure of a molecule, a CRN is probably not the right modeling framework. It could model the whole alpha helix and say, how does this helix interact with something else? But it's not going to be able to model the detailed 3D conformation. So finally, before I'm done with sort of the introductory part of this, and the background is I want to talk briefly about rate functions. These are very important. It looks like I might have a question.
You do. Can you see it while you screen share?
Yes. So the question is, what is the biggest system I can represent as a CRN? Could you do whole metabolism? Yes, absolutely. And I mean, I have simulated CRNs that have, I don't know, 100,000 reactions on my computer. They're purely of mathematical interest. You could easily simulate all of E. coli's metabolism, which has, I don't know, maybe 5,000 or 10,000 reactions, depending upon how you model it. Not a problem. You'd probably want to use a computer cluster to do that, or at least a very fast workstation. But yes, you could do it. And I think the biggest problem is figuring out the kinetic parameters. So on the function of kinetic, or on the topic of kinetic parameters, these are the things that parameterize your rate functions. This is where sort of your knowledge and experimental data will come in. And so the most common rate function you might use is mass action, and this is given by this equation here. And this, you know, if you're not used to products like this, we'll just go through an example. So if I have nothing goes to A, my rate is K. That's pretty simple. If I have A goes to nothing, my rate is K times A. So I'm taking the stuff on the left and multiplying it by the rate, and that's the rate that I produce the stuff on the right. If I have C plus 2B goes to C plus A, well, my rate here is KCB squared. So I have B squared because I have a 2B here. So the coefficients on the left become exponents in the rate. And that's mass action for you. And this is stuff that you maybe saw in high school or college intro chemistry. I generally think of mass action rates as rooted in statistical physics. These are true rates of chemical reaction in sort of the well-mixed formalism of chemical reaction networks, but many biologists use additional rates as well. And when using non-mass action rate functions, you really should think of them as approximations or model reductions to represent larger mass action CRNs. And so a very common example of this is the Hill function. And Hill functions make these nice little sigmoid shapes. So here I've shown a Hill function. This is the mathematical form, and I'm varying n, or in the first one, I'm varying k, which determines my inflection points. Sorry, my maximum value, little k. In the second one, I'm varying big K, which determines my inflection point. And in the third one, I'm varying n, which determines how steep the Hill function is. So these are very useful equations. They come up in enzyme kinetics. They can be used to model gene expression. But again, they're approximations that have certain regimes of correctness and other regimes are not valid. Another example is a negative Hill function, which is very much like a Hill function, but reversed. Instead of going up, it goes down. So let's now get into sort of the meat of this talk. We've described what CRNs are, that you have these rate functions, these big networks. I'm going to show you some examples and talk about how to tame the complexity of large-scale biological models with Biocompiler. So let's first sort of give an example of this complexity. So here is a little cartoon picture of a chemical system that many of us might be familiar with, where maybe we have, or an abstract chemical system, we have an external signal that diffuses into the cell. That signal binds to some transcription factor, activating it, making it go from F0 to F1. That transcription factor combined to a gene. When the gene is bound to the transcription factor, it recruits a polymerase. The polymerase allows the gene to transcribe and transcript or RNA. The RNA can then bind to a ribosome, which can be translated to produce a protein product X. And finally, because we're in a cell, the transcript, the signal, and X are all degraded via dilution as the cell grows. So this is a very simple model of what, the type of thing that synthetic biologists think about all the time, and maybe some other people as well. And I can write this down, right? This is not too complicated. In fact, I'll write it down in Biocompiler code for you right now. So I can create all the species. Here's S external, and I'm using these attributes, just say it's external. But again, this is just sort of code. It doesn't matter that much. Here's the species S. Again, it's different from S external because it has no attributes. Here's my species F naught. I can form complexes of species that are bound together using this complex function. I can have my complex or my gene, which I'm giving a material type DNA, but I'm giving it the same name as the protein. And I can go on and do a bunch more of these to get all of the species you just saw. I then provide some rates. Here I'm only using 5 rates to make it very simple. And I can go make reactions. Excuse me 1 second, because Slack is going off like it's nobody's business. So how do you make reactions? Well, I take my list of inputs on the left and my outputs on the right, and I have my rates. And this is a reversible reaction for S going from external to internal. Or here's another one where I have S binding to the transcription factor. And here's all the other reactions. There's 10 of them in total. So I could write this down by hand pretty easily. I then compile, I then put it into a CRN. And that's pretty straightforward. You could also imagine writing this down as an ordinary differential equation. So this one in particular would have 39 terms and 12 variables. So that's, okay, a little bit big, but many people work with stuff this large by hand, and it's not a big deal. But it's really easy to get bigger. So if I take my original model, I can just say, well, this is actually really simplistic. I want to add more details. First of all, F, the transcription factor, is actually being produced by a gene that encodes for it, which is maybe regulated by something. So here's just 6 more reactions, some of which are reversible, to encode for the production of F from a gene. Well, I might also say, well, actually in a cell, there's also RNases. The RNAs are degraded differently from proteins because there's little enzymes that go around and chew them up. So here are 4 more reactions to include the degradation of my 2 transcripts by RNases. And I can keep adding more things. I could add more transcription translation machinery like sigma factors in E. coli. I could add energy sources like ATP or some sort of energy metabolism, Krebs cycle, whatever I want. I could add multiple occupancy of polymerases and ribosomes, where more than one ribosome binds to a transcript. I can add background cellular processes like the production of ribosomes. Was that a question for me?
I think it was just a comment that
Okay.
you were cheating by preparing text instead of writing live stream.
Yeah, I'm working on my Blackboard skills over Zoom someday. So this gets very complicated very quickly. As soon as we have all of these additional terms, you probably wouldn't want to write this down by hand as ODEs. You could still write down all 20 of these reactions if you wanted, but now I'm getting even more complicated. It would be nice if there was a simple piece of software that would help you manage these growing models that are increasingly complicated. Or what if I have this module that represents the system I made, and I want to combine it with a module that represents the system my colleague made? Well, now I've just doubled everything. And that gets, you know, possibly quite challenging to model. So compilation is a way around this. We can, instead of writing down everything by hand, we can write down an abstract specification and get a very detailed implementation. And this is something that many of you might be familiar with in terms of computer code. C++ takes this, computer code you write and compiles it into machine code or binary that your computer reads. And importantly, the same C++ code can be compiled in multiple ways. So if I compile C++ on an Apple OS versus Windows versus Linux, I actually get different binary files, even though I start with the same code that does the same thing. So this is sort of the notion of compilation, is that it's a high-level specification that can become an implementation, but it doesn't become a unique implementation. It can become multiple equivalent implementations. And I've actually been talking about compilation all along here because chemical reaction networks are a form of low-level model compilation. So I start with these arrows and symbols, A goes to B, and I compile that into either deterministic ordinary differential equations or the stochastic equations I showed you. And this is a very simple level of compilation that even isn't enough for the level of complexity we deal with in biology. We want something higher level, and there's a lot of higher level compilation in biochemical modeling. So I'm just going to do a quick review of the literature here to say that I'm not the first one to do this, although I think Biocompiler is unique. A lot of my inspiration comes from the DNA strand displacement community, where they've invented many compilers. The most common type of compiler they have take CRN models. So I have a chemical reaction network I want to build out of DNA strands, and my implementation is what DNA strands do I actually use. So this is a CRN to CRN compiler. There's also some programming languages that have been written that take some abstract type of text, like C++ or something that looks like it, and produce DNA strands that implement that language. And finally, there's the notion of reaction enumeration, which says if I have sort of this description of the domains of my DNA strands, here's how my, here's the sequences I have, can I generate all of the possible reactions and ways they combine together? There's another type of modeling called rule-based modeling, which is used a lot in systems biology. I think BioNetGen and Kappa are the most well-known pieces of softwares or frameworks. And these are really good at dealing with combinatoric complexity. So if I have, say, a piece of DNA that has lots of binding sites on it, there's an exponential numbers of conformations that DNA can be in because of all the different binding sites. So BioNetGen and Kappa were designed to sort of deal with that. And then finally, there's some context-specific compilers. So there's integrase circuits that can be compiled. A spiritual predecessor to Biocompiler is the MATLAB, what do they call it? TXTL sim toolbox. And what these do is these are much more like Biocompiler in the way they're used. They sort of take a high-level specification and produce CRNs, but they're very limited in scope. They only deal with integrases or only deal with cell extracts. Biocompiler takes all of these things and combines them. It tries to be a unifying framework for all the different types of compilation I just showed you. And on one hand, it's a library to take simple specifications and produce complex implementations. And so that library consists of a couple things. It takes mechanisms, which are biochemical processes like transcription. It takes components, which are biochemical parts like a gene. And it takes mixtures, which are context. And these could be something like our biochemical context, like cell extract, or it could also be a modeling context, like we're going to assume ribosomes are constant. And it combines these three things to get a chemical reaction network implementation, which is saved as systems biology markup language, which is a common way to represent chemical reaction networks that can be simulated with many different simulators. So the idea here is that Biocompiler should fit into your existing pipelines if you have them, or if you already have simulators you like using, you should just be able to use it on top of whatever you do already. So besides being a library, it's also an object-oriented framework. So it should be easy for you to extend Biocompiler to your own needs. And so here is a picture of the class structure of Biocompiler at a high level, and I'm going to break this down because it looks like a fair bit, looks a little complicated, although as far as software goes, it's actually not too bad. So let's first start talking about mechanisms. So mechanisms are really the heart of Biocompiler. And mechanisms represent biochemical processes. So for example, I might have a transport process where something from inside goes to outside or vice versa. Or I might have binding. So here I'm having two different binding reactions where 2s binds to F0 or F1 binds to G. And these are both sort of the same motif. The equations look very similar. Or I have transcription. Here I have two different transcription modules. Transcription includes binding, you might notice, but it's more than just binding. It also has this polymerase, which produces a transcript. And then I have translation, which looks a lot like transcription. I have dilution. And finally, I have enzymatic degradation, another module. So this allows me to factor this very complex CRN with 20 reactions into 6 different motifs that are repeated over and over again. So in some ways, this makes it much simpler to imagine, if I wanted to write down, dilution happens in all these species, translation happens in all these ones, transcription happens in all these ones. It lets you simplify the way you describe it, perhaps compared to writing down all 20 reactions. But even more importantly, mechanisms are black boxes. So not only can I say transcription happens in all these species, but I can change how transcription happens programmatically. So for example, with transcription, there's sort of one unifying framework. The input is a gene and the output is a transcript. But what happens in the middle is programmable. So I might have a very simple model, like gene goes to gene plus transcript. It can't get any simpler than this. But maybe I want to include some machinery like RNA polymerase. So I have this binding reaction I've shown you a bunch of times where the gene binds to RNAP, and then the complex of the gene and the RNAP then produces a transcript. Or maybe I want to have a different version of something very similar where the gene produces a transcript, but now I use a Hill function instead of 2 reactions. So now I have this positive Hill function with my maximum rate being the amount of RNA polymerase. Or maybe I want to actually include multiple occupancy where a gene can bind to multiple RNAPs at the same time. These are all valid transcription mechanisms. And as soon as I say that the gene G is transcribable, with Biocompiler, I can very quickly change which one I'm using inside of my model. So that sort of covers what mechanisms are. The next major class in Biocompiler is the components. And components are what you might think they are. They're the things that do the mechanisms. For example, a signaling component is likely the thing that is going to be involved in transport. For binding, we have something called a chemical complex to represent a bound complex of things. For transcription, well, that's guided by a promoter. Translation is guided by a ribosome binding site and maybe the CDS, the coding sequence. And enzymatic degradation is really due to this RNase that can find all different RNAs and degrade them. So components are the things I'm saying I have these components inside of my system and they have different functionality assigned to them. And importantly, components can be hierarchical. So here's a plasmid. You'll notice that it has, well, let's show you the S-fold glyphs if you're not familiar with them. So this is in Biocompiler, we call this a DNA construct, and it's made out of DNA parts. And it has a promoter, and it has CDSs, and it has ribosome binding sites or ribosome entry sites, and it has terminators. And each of these codes a different functionality. So the promoter is going to show transcription. As soon as we see the promoter, we're going to say, oh, there's a promoter, and then there's a terminator. We know that we can transcribe these 2 different RNAs. And in this case, our promoter has information that is repressed by TEP-R. So now we know that this transcription is repressible. Then we have translation. We see that we have ribosome binding sites and CDSs, so we can reduce CFP, GFP, and RFP. And finally, we want to put all of this stuff inside of a context. So here we have a complex parts that perhaps generates some other parts and does a bunch of different processes, transcription and translation. with many different subcomponents. What is the mixture? Well, the mixture is the context. And for example, we can compile this in a simple mixture. So here I'm using the simplest modeling framework I can imagine, where everything's catalytic, there's no machinery, and I get this very simple chemical reaction network. And here the chemical reaction network I'm showing in this sort of bi-graph formalism, where we have two different types of nodes. The square ones are reactions and the round ones are species. And so you'll see here that we have our two different RNAs, which are both involved in a couple of reactions, and our DNA, which is also involved in a few reactions. Or I can compile this in a much more complex mixture, where here I'm including a lot of machinery. So I'm including things like RNA polymerase and ribosomes. And that gives rise to many more reactions that the DNA and the RNAs are both involved in. But importantly, I'm taking the same specification, this piece of this plasmid, and I'm just putting it into different contexts that have different mechanisms attached to them to get two very different models. And this is as simple as changing one line of code to go from the simple mixture to the complex mixture. So it allows you to take a system and ask, in a very idealized sense, how will it behave? And in a more realistic sense, how will it behave? So let's put this all together briefly. We start at the high level with the mixture, which has components inside of it. These might automatically be there, or maybe you add them yourself. Components and mixtures can both include mechanisms describing how biochemical processes work. If a component doesn't have its own custom mechanism, it will default to the one in the mixture. Mechanisms produce species and reactions, which when combined with parameters produce a compiled chemical reaction network. Those parameters, again, can belong either to a specific component or to the mixture as a whole. So there's different places you can put them. Finally, there's component enumeration, which I'm not going to talk about, but it's a way for components to produce more components. This makes this into a sort of universal compiler. And there's global mechanisms, which is a way for mixtures to apply mechanisms. And in the end, you get a compiled chemical reaction network. All of these things are objects that you can subclass and customize, and we have a whole library of ones that we think are useful for biologists, and so far, very few people have had to write their own. So I'm going to make a couple points about Biocompiler. The first is that it's modular. I can start with one piece and I can reuse it. So I can generate lots of architectures by reusing components. So for example, this is what we call a DNA assembly. It's a very simple motif of a promoter that produces something via transcription and translation. And if I take one of them and use it to repress another, I get sort of this inducible repression. Or I can take two of them and wire them negatively against each other and get a bistable toggle switch, as many of you have probably heard about this circuit. And here I'm showing the phase portrait. Depending upon my initial condition, I go to different attractor regions. Or I can take three of these and wire them into each other and get with repression, and get a repressilator, an oscillating circuit. So again, the same component, this DNA assembly, has been rewired in many different ways to produce many different architectures. I can really explore design space this way. Biocompiler is also expressive. I can explore circuit implementation. So if I was exploring design space in the previous slide, now I'm exploring implementation space. What does that mean? Well, if I take an idealized inducible repression, I say, okay, well, the repressor is repressing the reporter. How might I actually build that in the lab? Well, maybe I do it with a deactivated Cas9 guide RNA implementation, where now I actually have two things. I have a guide RNA and a dCas9 that are both expressed. They bind together and bind to the promoter of this reporter and repress it. Well, now it's a little more complicated. And if I actually look at how my induction behavior behaves, it's different. So in this case, on the bottom, I'm titrating the amount of guide RNA. And on the top, on the left, I'm titrating the amount of the dCas. And you'll see that you actually can get upregulation if you have a small amount of guide RNA and no dCas, or if you have a small amount of dCas and no guide RNA. But if you have a lot of both, you get repression. And this is something we actually see in the lab. If you build these systems in the lab and induce them in different ways, we can see this retroactivity where something that should repress is actually sometimes causing activation instead. And what's really happening here is that the production of transcripts is sequestering away the RNases that would otherwise be degrading transcripts of the reporter. So by having less degradation, you get upregulation. Similarly, with the transcription factor toggle switch, I can take this sort of idealized model and I can ask, well, maybe I could build it in a different way. Instead of having transcription factors repressing each other, I can have do it at an RNA level where I have these CDSs are coding for targeted RNases that degrade the other RNase. And this is a hypothetical model. No one's ever built this, but it looks like, at least according to these simulations, that you could have a bistable toggle switch at the RNA level. And I'll get to this question in 1 second. So finally, Biocompiler helps manage complexity. So this lets you test modeling assumptions and simplifications. If you look at the repressor layer, this is very idealized. In actuality, if you look at transcription, say under this electron microscope, you'll see that there's many ribosomes bound to any given transcript. So I can ask, if I make the same model of the repressor layer, but with multiple occupancy where many ribosomes can bound to one transcript, do I still get the same dynamics? Well, they're a little different, but I still oscillate. So it's showing that the simplifications made in this very simple model with no ribosomes are actually not terrible compared to a model that has, a very high occupancy of ribosomes in the transcript. Briefly, Biocompiler manages parameters. This is very useful because it lets me say I can have very specific parameters for every single reaction in my model, or I can use, say, 5 or 10 parameters to make a very big model. It will still work, and it still compiles by using these default parameters, and you can then fill them in as you get information. This lets you build models much more quickly and get prototypes up and running without having to do detailed literature search. And coding with Biocompiler is easy. This is just some code. It's a couple lines to produce the idealized transcription factor toggle. This makes 6 species and 10 reactions, so not very much. If I want to make the more complicated one, it's only slightly more code. And I'm going through this very fast because I'm realizing I'm running out of time. And ultimately, what I want to say is that Biocompiler makes model building efficient. So if you look at all the models I've shown you, the small ones might have, only a few reactions and species and only take a couple of lines of code to create. But for the very large models on the right-hand side, where you have, over 24 species and 36 reactions and many more terms in your ODEs, it takes, roughly speaking, the same amount of code to generate these larger models, but they're hugely more complicated. So Biocompiler really lets you generate very complex, sophisticated models without using a ton of coding. So Biocompiler is stable, extendable, and user-friendly. You can pip install it if you know what that is. It's all on GitHub. You can download it there and run it from there as well. We have automated unit testing to make sure everything is stable and extensive example notebooks. And as a quick summary, Biocompiler is software to compile complex models from high-level specifications into more complex models. So lots of complex models there. CRN compilation makes modeling efficient, modular, and reliable. I want to stress this. This is not something a lot of synthetic biologists or systems biologists are using, but I think it should become something that we all use to streamline our model building process. And the way Biocompiler does this is using mechanisms to represent biochemical processes, components to represent biochemical functionality, and mixtures to represent biochemical and modeling context. Parameter defaulting is super useful because it lets you build models before you've identified all your parameters, so you can just use sort of these default guesses. And Biocompiler can be easily extended and customized if you're a developer for your own modeling needs. It's not hard to use. It's been engineered for developers as well as for bioengineers and people who just want a script. So thank you to the other developers. Zoltan Tusa from Imperial, Andre Schur, and Ayush Pandey from Caltech. Richard Murray has been very supportive. The tons of TAs and early adopters and testers who made this possible, and the NSF, who's funded much of this project. And a brief shout out to the Build a Cell modeling series. If this was interesting to you, I would encourage you to look at the Build a Cell YouTube channel. There's this modeling series which goes through the whole design, build, test pipeline. We start with a couple different pieces of software to design models. We talk about simulating them, and we talk about inference. And then we also talk about scaling up multi-scale simulations with Varium. There'll be a talk on that next week, I think, in this seminar series as well. So highly encourage you to see that. It's very cool software. And this is great if you're trying to learn how to do this stuff. So, I've given you a high level overview here. These tutorials are have many more details, and as well as coding demos, so you can actually see how coding. And to get started, those are some options. Now let's take some questions. So let's see. I have a couple of, yeah, can I go back through the chat first? Is that okay?
Okay.
Exploring implementations, how is the functionality for programmatic CRN to CRN transformations? We have a little bit of that, but not very much. I think that is a place where we could add functionality. But again, we're not trying to transform CRNs. We're really trying to compile them. So our way of doing CRN to CRN implementations is or transformations, say, okay, if I want to change a mechanism that represents translation or transcription, I can change that and then recompile. So we're not so much as a CRN manipulator as we are a CRN compiler. So is Biocompiler open to code contributions? Absolutely. If you want to contribute to this and be part of it, we would love to have you contribute. Look at our GitHub, there's issues, and talk to us. And we would love to collaborate. We're actively collaborating with lots of people. Can you do parameter sweeps? So Biocompiler is not a simulator, it is a compiler. So if you want to do parameter sweeps, you probably want to do that via your simulator, although you could compile a bunch of models over and over again and change one parameter if you wanted to, but that's probably a little slower than changing it via your simulator. But what I will say is that when Biocompiler produces a model, it can give a parameter ID to each parameter you want, so it's very easy to access that via any SBML simulator to do a sweep. So it's set up so you can do those sweeps with this SBML simulator. Encapsulation. So encapsulation is an interesting question. We do have some basic level of compartments in Biocompiler. We don't, volume, again, Biocompiler isn't a simulator, so volume is something your simulator is going to have to take care of. But if you had a simulator that could deal with multiple compartments, and these do exist, and they could then have multiple volumes. So I'm not sure if that really answers your question, Chris. And I'll just stream through these and then we'll open it up. Alex says, is there a way to easily do things such as plasmid copy number changes, which change multiple? Yes. So you can change the initial condition of anything. So if you want to change your plasmid copy number, in this case, you would basically think about that as changing the initial concentration of a plasmid, would be one way to do it. And you can do that. Initial conditions are parameters in Biocompiler. So it will search your parameter databases that are parameter files, and you can also overload initial conditions with the parameter dictionary. So yeah, any more questions? I know I went kind of quickly at the end there because I saw I ran out of time. Great, well, thank you everyone for listening. Oh.
I think you had someone who wanted to ask a question who unmuted himself, and I don't see him now.
Yeah, I feel like he might have typed his question.
Yep. Okay, so thank you so much, William. That was a really great introduction.
Yeah, and please everyone, if you're interested in using the software, feel free to reach out to me over, say, the Build a Cell Slack. If you want some tips and tricks and help getting started, we're happy to help you figure out how to apply it to your modeling needs or direct you towards better pieces of software if ours is not the correct one.
Thanks, everyone, and have a great week. Bye.